Diffusion is the movement of particles from an area where they are in high concentration to an area where they are in low concentration. Let's look at an example. When we are baking cookies, the scrumptious smell of the cookies is high in concentration around the oven. Over time, that smell wafts through the entire house. The smell particles move from an area of high concentration to an area of low concentration. This is diffusion. Osmosis is a special kind of diffusion that involves only one kind of particle, water molecules. During osmosis, water molecules will travel across a semi-permeable membrane from an area where they are in high concentration to an area where they are in low concentration. In other words, they will move from a dilute region to a concentrated region. Let's look at an example. Let's say a tube is filled with water and the right and left halves are separated by a semi-permeable membrane. One side has lots of sugar molecules and the other side has fewer sugar molecules. The side with more sugar molecules is concentrated and the side with fewer sugar molecules is dilute. Water will travel from the dilute side to the concentrated side. This happens so the system can reach equilibrium and the concentration becomes equal on both sides. Osmosis is fairly straightforward, however, students often confuse which direction water should be traveling in. It's a good trick to remember that osmosis is just like diffusion, where the particles go from where they are more in number to where they are less. Similarly, water travels where it is more in number to where it is less. Let's test our knowledge with a question we see often in biology exams. Let's say there is a bag filled with 25% sugar solution immersed in a beaker with 50% sugar solution. Note that the bag serves as the semi-permeable membrane. Which direction does the water travel in? The first step to answering this question is to figure out which solution is concentrated and which is dilute. Is 25% sugar solution more concentrated or 50% sugar solution? The 25% sugar solution is dilute and the 50% sugar solution is concentrated. Why? The 25% sugar solution has 25% sugar and 75% water. The 50% sugar solution has 50% sugar and 50% water, making the 25% sugar solution dilute since it has less sugar than the 50% sugar solution. It's important to keep in mind that we're figuring out which solution is dilute and concentrated compared to each other. Now, remember we said that during osmosis, water travels from a dilute region to a concentrated region. So it'll travel from the 25% sugar solution to the 50% sugar solution. The goal is to make the concentration equal on both sides. Tonicity is a term we hear when we discuss osmosis. Tonicity describes how an extracellular fluid concentration can change a cell's volume by affecting osmosis. Remember that cells are surrounded by fluid, which is called extracellular fluid. As compared to the cell, the extracellular fluid can be hypertonic, hypotonic, or isotonic. Hypertonic refers to a really concentrated solution. Hyper means to have too much of something. In this case, it would be too many solute particles like those of sugar or salt. Hypotonic refers to a dilute solution. Hypo means to have less of something. A hypotonic solution has less solute particles. And an isotonic solution has equal amounts of solute particles as compared to the solution around it. In our example earlier, the 25% sugar solution would be hypotonic and the 50% sugar solution would be hypertonic. Let's look at our red blood cell to understand osmosis in our body better. In this picture, we can see a red blood cell surrounded by three different kinds of environments: hypotonic, isotonic, and hypertonic. The environment is considered hypotonic in comparison to the cell. Water will enter the cell, which is more concentrated as compared to its surroundings. This will eventually cause the cell to burst or lyse. An isotonic solution means the concentrations are equal on both sides, inside and outside the cell, and so there will be no net movement of water. This is usually how our cells exist. In a hypertonic environment, water will leave the cell, causing it to shrivel. The cell itself is hypotonic as compared to its surrounding. Water always travels from a hypotonic or dilute region to a hypertonic or concentrated region. The key takeaway in figuring out which direction water moves in is to determine which solution is hypertonic and which is hypotonic. Once we understand that, we can easily predict which direction water will travel in. The reason we study diffusion and osmosis in biology is because these processes constantly occur in the cell. Water and other molecules like glucose, sodium, potassium, oxygen, carbon dioxide, etc. are constantly entering and exiting the cell through diffusion and osmosis. That's all I have. I hope you found it helpful.